to start.
All right. Welcome to Mathematical Biology Seminar. It's the first seminar of this semester. Today's speaker is Calina Copos. She's an assistant professor at the School of Science at Northeastern University. Her research focus is on mathematical biology with applications to cell motility and cell cytoskeleton. And it's our pleasure to have her. Let's welcome her.
Thank you so much. I have to admit, I was mentioning this earlier, I've been meaning to visit Brandeis University for the last year since I moved to Northeastern. And I thought this was the perfect little push or excuse for me to finally come visit. And I'm so sorry that things did not work out. We were supposed to have our, I'm in the graduate committee, and we were supposed to have our new graduate welcome on Friday. And I'm not sure what happened. Well, but by emergency, we had to move it today. So I have to be on campus and meet with the new graduate students and say hello. So that's the reason I'm not at Brandeis, but still fall semester. The fall semester is we're pretty early in it. So I hope to be visiting very soon. There are a lot of people in both mathematics and physics' work is complementary to mine, and I follow it. So hope to see you soon. So as was mentioned, I am at Northeastern University. I'm split between the biology and mathematics department. I run a dry lab. That means all we do are computations and theory. I am hiring and very actively recruiting. So I'm happy to say a little bit more about that as well. So thank you for the opportunity to speak today. Not only am I thrilled to connect to people in the Boston area, but also selfishly, I want to tell you about a story, partly published, partly not, to gauge your interest in it. So today's talk will be about Scallop Theorem, which comes up in fluid dynamics. And I'll point to its relevancy and things I'm interested in, like cell locomotion. So before I start, let me say that this work was partly done with Wanda Strachowski. She is an associate professor at Case Western Reserve University. We recently wrote a paper and published it together, and we're gearing up to, I'm saying it so that I commit to it. We're gearing up for our second paper this fall semester. And this is a platform for me to kind of test these ideas. So thank you for being my guinea pigs here, and just kind of seeing how interesting this is. And before I move on anymore, small group of people, ask as many questions, feel free to post them in chat. Unmute yourself, interrupt me at any point. Nothing is more important than to have a conversation and get your ideas here. So I trained as a mathematician. Actually, I trained as a physicist and a mathematician. And it was novel to me when I started this that cells move. So cells move. There are good examples in healthy states of the human body. So for example, in wound healing, when a gap or a scratch is formed in the epithelial tissue, cells will slowly move in to close that gap. So that's one example of cell locomotion. Another example is when the immune system responds to any invaders into the scar tissue. So for example, this is a white blood cell or a neutrophil that's chasing a microbe here. And what I want you to see is the plasticity or the flexibility of the neutrophil of the white blood cell as it chases this microbe, but also its stiffness and rigidity in order to push away the red blood cells and platelets. There are also examples in disease states. A classic example comes from metastasis. So this is when a single cell or a group of cells leave or detach from a primary tumor site, enter the bloodstream, later exit the bloodstream and form a secondary tumor site. What you're looking at here, stained in blue, is the cell boundary of a metastatic cancer breast cell. And in orange are fibers that make up the exterior, the exterior tissue that this breast cancer cell is invading. And the picture suggests that the cell is squeezing through the gaps in order to move or propel forwards. And as we were just speaking earlier, the direction in this field is no longer to focus on single cell locomotion, but collective migration, and we do that in my group as well, the collective behavior of cells, and we're interested in embryonic development. So what's the status quo of cell locomotion? So for the last, I want to say century, but more like 80 years, what people have done is taken cells, put them on flat surfaces and watch them under the microscope migrate. And a number of experiments, drugs that were given to the cells, other perturbations, careful theory built together with experiments have led us to the following mechanism of cell locomotion. So a cell is placed on a surface. It's rather symmetric. And the first thing it does is it forms cell surface adhesions. These cell surface adhesions are bonds. They're made up of proteins. And what they do is they allow the cell to transmit its internal forces to the exterior. In other words, it generates traction. The next thing that the cell does in order to migrate is it breaks its symmetry. How it does this is also something that has been studied together by mathematicians and biologists. There are many ways that this can happen. One way this can happen is through cues from the environment, from the exterior. Those cues can be mechanical, chemical gradients, or even electric fields. So cells even move in the presence of electric fields. Another way this can happen is spontaneously. But whatever the means it is, this causes the cell to elongate in a certain direction. That's the direction it wants to move in, and it forms what's called a protrusion, an elongation of the cell boundary. This elongation then needs to adhere to or stick to the surface underneath. So new adhesion or bonds or links form between this new cell extension and the surface underneath. And finally, now the cell has achieved its maximum length. It's lengthened, it's attached. In order to translocate, it needs to break the adhesions at the rear. That causes the whole cell to translocate forward. And then this cycle of locomotion is repeated over and over again.
I have a quick question.
Absolutely.
Is there one determinant factor that decides the velocity of the cell locomotion?
Many. I don't know if there's a single one. Yeah, they're a very good question. There are many determinants. Shortly put, everything from the cell type to the presence of cues from the environment. And you should be asking not just about its velocity, but its persistence, right? You can imagine kind of wobbling around. So there are many environments, from exterior environments, mechanical, chemical environments, to interior modes of migration and so on. This lovely cell you're seeing here has been studied for a number of years. It's called a keratocyte. It's collected from the fish outer skin layer. And it moves very quickly. It moves, I think, like a body length every second or so. Very fast moving cell. Oh, of course, and another thing that really determines how quickly the cells moves is this the relationship between the bonds that are formed between the cell and the surface. It's a big determinant too. But of course, this is a very simplified version. In the last 30 years or so, people have started with improvements in microscopy. People have started being able to image cells in living tissue and living organisms. And they saw, okay, that this, of course, we knew that this was fairly simplified to look at a cell on a flat surface, but it turns out not just the richness of the environment is different, but the way the cells move. So we learned that there's this existing paradigm of moving on a flat surface does not apply very well to cells moving in more complicated, physiologically three-dimensional environments. And to kind of convince you of that, let me show you what I call a confounding observation. These experimentalists that were more physics trained had this clever idea, and they said, it's really, really hard to image these cells in living systems. Let's try to mimic the environment of a living system by putting these metastatic, like breast cancer cells, in a channel. It's a microfluidic channel. There's background fluid, but no flow, so no pressure difference from one end to another. And let's do something else. Let's cover or coat the channel to make sure that the cell does not stick to the surface underneath, to the surface. So in other words, there's no bonds connecting the cell to the surface. And surprisingly, what they saw is that the cell appears to be doesn't appear. The cell moves, appearing to be using something like chimneying. So this idea of like wedging yourself into the gaps in order to propel yourself forward. And so when this came out, it perplexed people. It came out about, wow, I used to say 10 years now. I guess 10 years or so, a little bit more. It perplexed people how this happened. And there were a few verbal hypotheses that came out, including this idea. So why is it so perplexing? So if you think about it, to move, you need to somehow transmit your internal forces to the outside. The way we do it is through legs. The way cells do it typically is through bonds or adhesions. Okay, so you think maybe the cell, so maybe it's using pressure. Maybe somehow the cell is just pressurized, it exerts pressure onto the surface. Fine, you exert pressure, but if you exert pressure and your channel is straight, you're exerting pressure in the normal direction. In order to move in the tangential direction, you need tangential forces. So where are these tangential forces coming from? Okay, so then the hypothesis was, you know what, the channel actually is not quite straight. It has a little bit of give. It's hard to see it, but it has a little bit of flexibility to it. And so therefore, when it applies, when the cellular pressure applies those normal forces, then those normal forces actually have a bit of a tangential component. You can, I think this picture already tells you the what I'm going to object to here. Well, add up all of those tangential components. Some are to the right, some are to the left. They sure add up to zero. So how do you get net propulsion? How do you get consistent tangential forces in one direction? So that is the question that we posed. So if you were to sit in a lab where maybe you build robots or maybe you build a computer model, what would the necessary ingredients be in order to make a cell move in that channel without having any connections between the cell and the surface? So let me tell you a little bit first about the environment that that cells feel. So first of all, cells are relatively small. There are a few microns in length. Here you're looking at sea urchin, spermatozoa, one of my favorite bacteria, Helicobacter pylori, H. pylori, the cause of stomach ulcers and stomach cancer, and Paramecium, that has a bunch of flagellas on the exterior. So these cells not only are small, but they move slowly. At their fastest, they move maybe a few body lengths a second. Cells move even slower than that, or eukaryotic cells or mammalian cells that we focus on metastatic cancer. One thing you could do is you can compute what's called a Reynolds number. This is a dimensionless number used in fluid dynamics. It measures or compares quantitatively the inertial forces versus the viscous forces. It does so by taking a ratio of what appears here is the density of the fluid, the typical or characteristic velocity of the organism that moves through this fluid, the characteristic or typical length scale of the organism that moves through the fluid, and the fluid viscosity is on the denominator. And so you can plug in some numbers here for how big these organisms are, how quickly they move, the fluid density and viscosity, and you get a Reynolds number relatively close to 0. So what this suggests is that viscosity or viscous forces dominate over inertial forces. So one implication of this is that instead of Navier-Stokes' equations, what we use are Stokes equations, which are a simplification. Here are Stokes equations. The first one is a force balance. The second one comes from mass conservation and is an incompressibility constraint. So in the first equation, what you see here that comes up is mu is the fluid viscosity, P is the fluid pressure, U is the fluid velocity, and F are any external forces participating in the fluid. I'll come back to these external forces. Typically, they're not shown if you just look at Stokes equations. Only the first two terms show up. And the force balance says that pressure gradients are balanced by viscous forces. One thing about these equations that you notice is they're time independent, okay? And they are linear, both really nice things. Their consequences to this is that you can write nice Green's functions to these equations. And in a very nice paper, Purcell noted that any reciprocal motion cannot generate net propulsion. So he exemplified this through this cartoon image of a scallop. And he said a scallop opens and closes and opens and closes. Well, in Stokes' flow, if this scallop was very, very small and moving very, very slowly, the scallop would not go anywhere because of the reciprocability of, or the reversibility of the motion. So as the scallop opens up, it propels, and as it closes, it propels in the opposite direction, resulting in zero net movement. Same follows for an organism that has a rigid flagellum. And he proposed in this paper the simplest swimmer that can actually move forward in a Stokes regime, in a low Reynolds number regime. So I apologize about this typo here, but what are the implications for our system? So a little bit about our system. Our cell, again, is squeezed in this microfluidic channel. Our cell is about, okay, here we go. Our cell is about 10 microns, maybe 20 order of magnitude estimate. It actually moves considerably slower than flagella systems. It moves about a body length a minute instead of a second. Using the same fluid viscosity and density, let's pretend like this is just water. And so the Reynolds number that comes out is small, negligible. So the governing equations for our system are Stokes equations. and the system suffers of this Scallop theorem, if you will. If the motion is reciprocal or reversible, time reversible, then the system or the organism doesn't move forward. Clearly, this organism moves forward. So how does it do this? So the first question we asked is, okay, does it exhibit time irreversible shape changes? So here's how I think of a cell. I think of a cell as a bag of stuff. So this bag of stuff includes fluid, and it's in fluid. So a bag of fluid in fluid, or a balloon of fluid in fluid. And sure, inside the cell, there are many, many other things. The cell is pressurized, and there's a cell boundary. The cell boundary separates the exterior from the interior and ensures that the cell doesn't lose its volume and that it keeps everything inside of it, like DNA, RNA, proteins, and such. The cell boundary I labeled here as having coordinates X. And the system is governed by Stokes equations. So there are Stokes equations written again with incompressibility. So the cell doesn't want to lose its volume. The knowns of the problem are the position. So at all times, we know the position of the cell boundary. One thing we specify, and this is where modeling comes in, are the biomechanics. So what are the forces acting on the boundary? For example, you could say it's elastic, right? And that provides elastic resistance. Okay, so then the forces are, maybe you go with elastic springs. So the force is equal to K as spring stiffness times X minus X0, right? Or length, to be more precise, like norm X2, right? Like the length of every single minus the length, the resting leg. So knowing the positions, given a constitutive law or a law for the forces, how the forces depend on the position, you can compute those forces. Knowing the forces, you can then solve Stokes equations for the fluid pressure and the fluid velocity. So that is the statement of the problem. Once you know the fluid pressure and the fluid velocity, you can move the boundary. The boundary moves at the same speed as the fluid pressure. Now that you've moved the boundary, you know the new position of the boundary, you compute the forces, you compute the fluid velocity and pressure, you move the boundary with the fluid pressure, with the fluid velocity, and continue updating the system in time. A little bit more about the system we want to model. So it's a little bit more complicated. So first of all, we'll talk about the channel or the environment, but first thing is looking at these figures or this movie over here. Let me stop it because it's a bit too fast. The cell moves using pressure. So if you look at the very, very front, pay attention in this frontal region, you'll see these bubbles forming. Let me play it again. So look for these bubbles. Yeah, hopefully I see a few nods in the crowd. Okay, so in the early 2000s, people paid a lot of attention to these bubbles that the cells were forming. Why so late? Again, people were just so focused on cells moving on flat surfaces. They didn't even, they thought that was the status quo. And actually, I don't have time to go into this, but for anybody who's interested, there's a really beautiful story about how people thought that cells moved on a flat surface, and they were like, let's build a drug that inhibits the cell's ability to form these adhesions to the substrate. And they got mixed. This is for cancer metastasis. They got mixed because they thought, hey, if we could stop cancer metastasizing, we could treat the primary tumor site, bam, done. But so they thought, great, the biggest problem here is the metastasis of cells. It's the motility of cells. So they thought, great, if we block the cell's ability to stick to the surface underneath, the cells won't be able to stick. If they can't stick, they can't move, they can't translocate, they can't exert traction. Problem solved. So they built this drug, and mixed reviews in mice, but there was so much interest and so much money poured into this problem that they decided to skip the normal stages of protocol for human testing, and they went directly into patient testing. I believe it's stage 2 or stage 3. I'm not very familiar with drug discovery, but what they saw is a much worse response. In fact, patients were dying at a faster rate than expected, and it took quite a bit of time to realize that what was happening is the cells, when their ability to stick to the surface was inhibited, they were using this squeezing mechanism, this pressure-driven mechanism to locomote, which actually ends up being a much more aggressive form of motility for metastasis. So that, and I'm probably doing a poor job of summarizing, but that's my understanding of this paper on the history of this drug. But that, together with other experimental observations, motivated people to kind of rethink this idea that there's only one paradigm, only one mode of locomotion, and paid more attention to what's happening in these pressure-driven bubbles. So what happens is, so that led to a few people studying these cells in simpler systems where they just put them on a surface and watch them undergo this pressure-driven expansion or protrusion. So it turns out what happens is that at the cell boundary, it's a bit more complicated, even more complicated than I'm showing you, but there is the cell membrane, which is this lipid bilayer membrane. And underneath, it's fairly flimsy and flexible and changes very quickly, but underneath it's supported by a thin layer of a polymer called actin, and this actin kind of gives it its rigidity. Not only does it give it its rigidity, but it provides contraction, and it's very tightly coupled by other polymers and other proteins to the bilipid membrane, and that's kind of what gives the bilayer lipid membrane it's the rigidity we see. Mechanically or mathematically or physically, whichever word you like to use, we can think of this as two elastic structures stapled together. Now imagine the system is in equilibrium. Now imagine you remove the couplings, the stapling between the two elastic membranes locally. Well, Well, that drives the cell membrane forward due to intracellular fluid flows, and that's what causes this expansion. Eventually, the expansion retracts because new material forms underneath, causing these two structures to be stapled back together. So let me see if I have a better, I'll have a better picture in a second. But again, the idea is that the stapling between, one way this could happen is the stapling between these two elastic structures is loosened or lost. On balance of forces, fluid flows. Fluid drives the membrane expansion, then the stapling reconnects, and the two elastic structures are again together, and they contract in to recover the initial shape. These are called fluid pressure-driven protrusions, or blebs. These are very different from what was in this paradigm status quo, which were actin-driven protrusions, for anyone familiar with these types of protrusions. Anyway, the important part here is fluid drives the protrusion. Okay, so in our model, we not only have the cell, but it's also placed in a channel. So it's placed in a microfluidic channel. There's no induced flow or background flow. There's no pressure gradient across the channel. The cell is modeled as these two elastic structures. One's called the membrane, the other one's called the actin-myosin cortex. I will simply refer to it as the cell boundary together. And the cell interior is just fluid. It's the same fluid as the exterior fluid. They have the same viscosity. Thomas has done quite a bit of work and is a master at solving structures in fluid, but he uses a slightly different method than what is here. He uses the immersed boundary method. It turns out these two methods are somewhat similar, yet executed differently. The method I use here is called the method of regularized Stokes slits. It takes advantage of Green's functions to Stokes equations. I was not going to show you this, but just to convince you there's math. These are the equations in all of their glory. So these are all the external forces. It turns out there are forces on the elastic membrane, on the cortex, in between the membrane and the cortex. Each one has its own constitutive law. Each one has its own force balance. And here are all of their constitutive laws. Nothing is super surprising. The membrane is described as an elastic contractile structure. The cortex is described as an elastic contractile structure. The membrane and the cortex stay together with springs. That's in red. And then, as I mentioned before, the membrane is impermeable. That means it moves with the fluid velocity, and the cortex has its own update equation for how it moves. So the velocity of the membrane and cortex are known once you know the fluid velocity. Okay, so now on to results. So again, our question is, these cells placed in a channel, cells cannot exert traction. That's not really the word I want to use, but there's no links, physical bonds between the cell and the surface. How does it push forward? How does it generate tangential traction, tangential forces? And how are those forces then transmitted to the surface? First thing we're going to ask is, does it break Scallop's theorem? It's an organism in Stokes. It's an organism in low Reynolds number. Are the shape changes that it's undergoing time irreversible? So first we remove the channel, and we just say the cell is undergoing this pressure-driven protrusion. In red, you'll see the initial outline. I'll show you a movie of this. This is the experimental movie. These are our computations. In here, what you'll see is the current position of the cell together with the initial position of the cell. So the cell starts symmetric, round, and with a radius of 5 microns. In black, yes, you cannot, the membrane and the cortex are so close together that you cannot visually distinguish them. So you'll just see a black contour that represents both of them. Dotted will be the old cortex as it reforms and breaks and reforms. And then in purple, white to purple is the fluid pressure. I visualize it here. And together with the arrows, which are fluid velocity or fluid flows. And I should say that this model was initially developed by Wanda Strachalski, my collaborator, and she published 3-4 papers that just try to get this as physiologically, as biologically as correct as possible. And so she did a lot of very careful work to make sure that this model is ground truth matches the biology. So we're fairly sure this is correct. So here's a movie of the cell undergoing protrusion expansion, protrusion retraction, protrusion expansion, protrusion retraction. And it undergoes about 6, I think more than 6 cycles, 8 cycles or so. In slow down, here's one cycle of the expansion phase. This is the maximum extension, and then finally the retraction phase. And your eyes don't deceive you. You might think, boy, that's fairly reversible. Sure enough, when we plot them right on top of each other, we see, yeah, this is a fairly reversible motion. So this is more or less, in its own way of expressing this, the Scallop theorem. So there's no movement because this motion is reversible. Okay, we have our answer. Scallop's theorem is not broken. The cell undergoes reversible time changes. Okay, now let's place it in a channel. Perhaps this is what it needs to break the symmetry. So we put it in a channel. We put it in a channel where it's squeezed so that it can exert that compression, that it's compressed in there, so it exerts pressure. The channel is rigid, it's not allowed to deform. And what we see is, again, cycles of forward and backward locomotion. If we take a look over a long time, so this is half of a minute, we see a displacement of the cell of about half of a micron. So that means over a full minute, that would be a displacement of about a micron. These cells are reported to move 10 microns a minute. So this cell moves about 10% of its length in a minute. We puzzled on why this is the case. We think this is because of volume loss that is due to the numerical method. So we think this is more or less numerical error. And it sounds like a lot, but 10% is about the same error in measurements. So we're pretty convinced that this is not significant locomotion. To test this further, we placed the cell in different confinement levels. So we made the channel either be a little bit wider or a little bit narrower, and the results stayed consistent. The cell barely moves anywhere. So no significant movement. The cell undergoes the same scallop-like movement, and we haven't quantified this, but this is not so surprising. Yeah, sure, it's squeezed. Yeah, sure, it goes forward and backwards, but we saw that it goes forwards and backwards about the same amount, and the squeezing just generates normal pressures in the normal direction, not so much in the tangential direction. So the quest is on. So we thought, what if we introduce topological asymmetry into the channel? What if the channel is not smooth? So we introduce what we call a serrated channel. The asymmetry is broken in two ways. One, top and bottom. The bottom is smooth, the top is serrated. We saw experiments that mimic this, so we wanted to mimic those experiments. And then we also, the serrated, we actually use a sawtooth function, and here it's asymmetrical in that it rises slower than it decays. So the slope on the rise is smaller than the slope on the decay in absolute value. And so we watch the cell, and the idea here the hypothesis is that maybe it wedges itself into these gaps in the channel. And yeah, it does. That's what we saw. Yeah, it does, until it kind of reaches an equilibrium. And then it's just stuck into this gap. And then we thought, you know what, let's change the frequency of the serrated channel, have either higher or lower frequency with different amplitudes so that maybe it can really get into that gap. And more or less, we saw the same result of, yeah, it maybe translocates a little bit, but then more or less it doesn't move. The motion is stationary. Yes. Yeah.
Sorry, I have a question. So,
absolutely.
Are those wedges allowed to penetrate the cell? Because they
very good. Yeah, very good question. Yeah, very good question. They're modeled as potentials. And so there is the potential, that potential has a constant there. So it is allowed, so it can, but there's a lot of force if there's a large penalty force if it does.
Okay, thank you.
Yeah, thank you. Good question. And I can also answer the question. Yeah, maybe towards the end, I'll answer the question of like, could you do it differently? Yes. And what would the problems be? And I think Thomas already knows very well what the problems are. Yeah, sorry. And I saw a question in Q&A. Is there a biological motivation for the sawtooth shape? Absolutely. Very good question as well. Yeah. Two reasons. One, there's literally a paper that came out after we published our paper that had a serrated no, I'm sorry. There's a paper that was published before our paper, but we only found out about it after we published our paper with a serrated channel. So in this paper, they said, look, it's an experimental paper, and they placed the cells in smooth channels, and they saw no locomotion, and then they placed the cells in serrated channels, and then they saw locomotion. So that is the one motivation. And then the other motivation is that we really wanted to get an elastic channel, something that allows the cell to wedge itself in. Turns out there's some numerical issues to do that accurately well. And so we decided that this would allow the cell to kind of wedge itself into the channel gaps without actually fully mimicking an elastic channel.
Sorry, I have one more question.
Absolutely.
So how do you pick the density of those wedges? Do you just like randomly pick a number, or is it actually biologically relevant parameter?
Oh, no, the way I built the serrated channel was completely random. Yeah, and this is partly why I think there's an initial displacement and that the cell is not quite right. You start with the wedge front being, you know, a little bit to the right, to the left, and so the cell maybe displaces it. But then it gets trapped, as you could see over here. It gets, or in these time snaps, it gets trapped in there and then just kind of exerts flows into that direction, and then it just gets stuck in that wedge. And so that's why you, that's why you see, for example, in this purple line or blue line here, a little bit of forward displacement, but then the cell just it's not moving anymore. Whereas you don't see it in these other simulations.
I see.
It has to do more with the initialization of how those wedges are than anything else. So surprisingly, we didn't see any significant movement in serrated channels either. That brought us back to the drawing board. So the motion is time reversible. Introducing geometry did not help. Introducing asymmetrical geometry did not help either. So we were kind of just trying to think of all the strategies that you could have for breaking Scallop's theorem. So one thing you could do is you could introduce a non-Newtonian fluid. I skimmed over this. Water is a Newtonian fluid. Mucus is a non-Newtonian fluid. For example, sperm is a flagella system, is a flagella flagellated, and it moves, it has a flexible flagella, and it moves in mucus, cervical mucus. So to appropriately model that system, sure, you really should be thinking about the exterior as a non-Newtonian fluid, but actually the viscosity of the cell exterior, depending on where you are in situations, is pretty close to a Newtonian fluid. So we didn't want to introduce a level of complexity when the complexity wasn't really physiologically motivated. Flexible structure. Okay, for example, this is how flagellas move. This is how sperm moves. It has a flexible flagella. Asymmetrical geometric structure. Okay, close to the boundary. We thought we were. And then a non-reciprocal actuation. So I'll come back to this. So flexible structure and non-reciprocal action. So we did what we could to the environment, we think. Sure, we could build an actual elastic channel, but there's experimental evidence that cells do move in serrated channels. So rigid serrated channels as well as just rigid straight channels. So those two experimental papers are kind of at odds with each other. Okay. So not really worth revisiting the boundary condition, but we did think a little bit about this flexible structure and non-reciprocal actuation. So what do I mean by non-reciprocal actuation? If you think about the scallop as this mechanism that opens and closes, and I think of it as two wedges connected by maybe an elastic spring. If the scallop does the same motion over and over again, it's a spring that opens back up to the same length, to that resting length, the cell doesn't move, or that scallop doesn't move anywhere in Stokes. But if instead maybe it doesn't open quite as much, well, now you're breaking the symmetry of the motion. There's a time-dependent history to the motion. Can we incorporate that into the cell? And so, yes, what we try to do is we said, okay, this boundary of the cell, perhaps it's not elastic and contractile, but perhaps it's viscoelastic. Perhaps by introducing viscosity, what I mean is viscoelastic response is the idea that stress relaxing, elastic stress relaxing over time, or put another way is that there's a history of deformation, of forgetting the deformation. Put even in a different way, it's the idea that you forget your reference configuration. So we can incorporate that. And let me just say for the biologists in the room and anybody else who's interested in the biological motivation of this, why? There's cortical turnover. There's a turnover of actin in the cortex, not to mention in the membrane, right? This lipid bilayer membrane is certainly not a static structure, and more so the underlying actin cortex. Cortical turnover, actin turnover is something that's fairly well known and accepted in the literature with timescales that are relevant for this locomotion, let's say timescales of 30 seconds. So what we did is we incorporated this into our model. So here is, I returned to this experiment here where we just have a cell undergoing these cycles of expansion and contraction of that protrusion. At the top is this elastic boundary, cell boundary, and at the bottom is a viscoelastic boundary. At the top, I'm showing you a cycle of expansion, maximal expansion, and then retraction. And here is 27-second represents 4 cycles after expansion and retraction. And the takeaway here is that if I look at 27 seconds at the top panel on the right side, ignore the fluid pressure, ignore the fluid flows, just look at the red and the black outline. It's really hard to tell them apart. They're pretty much lying on top of each other, meaning that the cell in 4 cycles of expansion and contraction did not move. At the bottom are 4 cycles, the same image shown to you after 27 seconds or 4 cycles. Lambda is the constant of time of memory. In other words, it's the longer lambda or the larger lambda it is, the shorter the memory, or sorry, the longer the memory. There we go. The larger the lambda, the longer the memory. So lambda going to infinity means perfect memory. I never forget my initial configuration. And sure enough, I couldn't use lambda is equal to infinity, mathematically speaking or computationally speaking. So I used a large lambda. I don't even remember what it was. And yeah, again, you kind of barely can see a difference between the black and the red outline. Whereas in the other ones where lambda, as you decrease lambda, so here lambda is 10 seconds, meaning that in 10 seconds, the material has forgotten its reference configuration, the cell displaces forward. Aha, we finally break Scallop's theorem. It's not a perfect way of describing this, and I'm trying to think of a good way of capturing this mathematically. But again, it's this idea that the scallop doesn't quite open as much, right? Why doesn't it open as much? Because it forgot what its reference configuration is. It forgot that this is no stress. Stress relaxes.
So, Calina, is one way
Yeah.
of thinking about this that once you add in this kind of turnover in the membrane, that kind of sequence of deformations that you showed us earlier that look pretty much, this bleb goes out, the bleb comes in. Now, is it kind of changing the this into kind of a power stroke and return stroke, where when the bleb goes out, it has like maybe one radius, but then because this this cortex is remodeling, when it's being withdrawn, that's sort of going through a different sequence of shapes. Just
absolutely, though there's no, as you would with a power stroke in a flagella system, you would kind of input that in some way into the return stroke, and here it's not. It's just it's independent of what's happening with the expansion and contraction. But yes, absolutely, the sequence of deformations is different. So the shape changes are different. Still struggling on how to quantify that to really show that, but yeah, that's right. So we see that there is locomotion with a viscoelastic boundary. Let's see what it looks like in kind of completeness here. So here I place the cell in a channel, now smooth channel. Now we see a cell speed of about 5 microns a minute, so half of a body length a minute. If we put a serrated channel, we see about 10 microns a minute. I'll remind you that this is biologically what's order of magnitude what's reported. In free space, the cell moves 3 microns a minute. So confinement certainly enhances migration speed. So the story, at least partially, is what do you need to move? You need viscoelasticity, so turnover, and you need confinement certainly enhances your speed of migration. And asymmetric geometry also enhances your speed of migration. The response is not so nice. The story is not so nice. It turns out it depends, these results depend very much on what I choose for my viscoelastic relaxation timescale. And it's a little bit of a different story if I choose a different relaxation timescale. But as long as the relaxation timescale is small enough, below 30 seconds, which is biologically right, that's the response. No, I don't understand what happens in this jump. But let me tell you about something that I see I'm kind of short on time. So let me tell you something that that is even more upsetting than this. So I said, well, how do these cells move without cell surface lengths? And so partly the answer is with viscoelasticity, with turnover, and with confinement. Smooth, serrated, either one does the trick. But that doesn't tell you the mechanism, right? That just tells you what you need in order to move. So it's a pretty unsatisfactory answer. We're not quite at the mechanistic step of lengthen, attach, detach, right? So there's still quite a little bit to be done here. So we're tracing through these different hypotheses. So one thing you could say, okay, so still you told me what you need in order to move, but you haven't told me how does the cell exert traction forces in the tangential direction to the environment. So one thing you could think of it is it's fluid flows. Okay, it's the extracellular fluid flows. Again, there's this thin layer between the cell and the surface. And that thin layer, because it's so thin and you have the cell moving back and forth, it's producing quite a bit of pressure between the cell and the wall. Perhaps it's the fluid flows coupled with the shape changes, and I'll come back to this in my next and last slide. And also, you know, a couple of you have asked about the extracellular or the channel. How is that modeled? Well, it's modeled using a penalty potential force, or coming from a potential energy. That's very different than saying velocity is zero there. Fluid velocity is zero on the channel. And the reason why we didn't do that is because then you have this actual place where the fluid flow is zero, whereas the fluid flow or the cell is moving back and forth just maybe a tenth of a micron away, causing these large pressures, fluid pressures. Now you have to resolve those flows, which are expensive to resolve and require not just high resolution, but also taking small time steps. So computationally expensive. Thomas has worked a bit on this and has good insights into how to do this. So instead, what we did is we didn't want to deal with all of this. We enforced zero flows further away from the channel so that we get away from this numerical inconvenience. And instead, we say there that, yeah, the cell just cannot penetrate past this point. Well, that induces a boundary slip. Is the boundary slip really what's causing this? Biologically, how do we feel about this? Okay, because actually the types of channels they use are pretty permeable. So we think that that's relevant. So lastly, I'll just leave you with this and say we did one more experiment here to kind of get at this idea. And what we did is we took out the fluid from our cell experiments. And we took out the fluid by saying there's no fluid pressures, the fluid instantaneously equilibrates. And when we did that, we saw no local motion. So somehow the fluid, the shape changes are different, and the fluid certainly plays a role in this. So we're tracking through these hypotheses, and I'm out of time here, so I'll just say thank you. We work on a wide array of problems, everything from cell locomotion to looking at experiments and measurements from experiments to a little bit of machine learning and learning about networks inside of cells. If you're interested at either the graduate, undergraduate, or postdoc level, I have funding and we are welcoming people in my group. So thank you so much and hope to see you later this semester in person.
Thank you so much. If anyone has questions.
Well, I guess just going back to the experiments that kind of motivated the work, how how similar are those experiments to some of the simulations you presented, where you kind of have like a repeated blebbing in and out of the cell? I kind of seem to remember when looking at those experiments, it's almost like there's sort of an initial bleb and then it just continues sort of translating. Maybe you could just say a bit about that.
Yeah, it appears that there's an initial bleb and then sequential blebs at that initial blebbing site. I think maybe you saw it a little bit in the movies, like multiple blebs. They're not quite all in the same position. The secondary blebs tend to be smaller.
And does that initial bleb ever really kind of withdraw?
It certainly does to a degree, but I'm not sure if it's a good question of whether they have the right resolution to kind of resolve that and see that. It's a really good point to take a look, and see if it's quite, if it retracts fully or not. That's a really good point. Yeah. I think this is, I think we partially motivated by experiments, and also when trying to do this as take away as much of the complexity as possible was our idea, to really try to get to how do you move using intracellular pressure in confinement. But it would be interesting to take a look at that, and I think we can. So
great.
I have a quick question.
Sure.
There was a story of single cell locomotion. Is there the same type of locomotion at the collective cell level?
Yeah, good question. Yeah, good question. I don't know is the answer. In my understanding from the experimental groups that I work with, you kind of choose your weapon of what do I mean by that? You choose whether you're going to do microscopy in vivo on an embryo, and then you don't really have that resolution to see the underlying actin structures, or you do these experiments in these more synthetic environments. Yeah, so I don't know of the systems I don't really know. It's yeah.
Okay, thank you so much. I think we have run out of time, so thank you, Calina, again.
Thank you so much.
Thanks, Calina.
Pleasure to
Thanks,
see you.
Calina.
Thank
Of
you.
course. Thank you.
And we'll see you in 2 weeks.
Perfect.
Yeah, bye.
Bye.